Hey guys, let's review just briefly from yesterday. What is a codon? So remember, a codon is 3 nitrogen bases found on the mRNA, and they code for an amino acid. Okay. And then the next part, what is meant by the term primary transcript? So the primary transcript, remember, is the piece of mRNA that is created just from the DNA. That's before it's modified. It's the rough draft, so to speak, of transcription. It's the piece of DNA that's just been copied into mRNA. And I talked to you a little bit yesterday about some of the processing that happens, but we're going to really go through that today in a lot more detail. But that's the pre-mRNA, or also known as the primary transcript. Okay, so yesterday we kind of looked at gene expression as an overview and how we do transcription from DNA to RNA, and then translation to go from that RNA to the protein. But today we are going to talk about transcription in a very detailed manner. So we're going to talk about the enzymes that are involved and just basically the steps and what happens exactly in transcription. So you've seen this enzyme or a close relative of it before. In DNA replication, like what happens before mitosis and meiosis when a cell's going to divide, you have DNA polymerase because you're copying DNA to DNA. Here, because we are making RNA, we have RNA polymerase. Okay, so don't confuse those two. DNA polymerase is in replication, RNA polymerase is in transcription. So RNA polymerase is our big enzyme here. That's the, we're going to talk about a couple of others, but RNA polymerase is really the manager of transcription. So there's several different types of RNA polymerase. In eukaryotes, they know of at least 3. Now, bacteria are thought to only have a single type of RNA polymerase, but there are a couple of different types. But in RNA synthesis, we have specifically RNA polymerase 2. Okay, but if you just know polymerase, then that's probably all that you need to know. Okay, so to get transcription started, we have a region on the DNA. Remember, transcription, we're going from DNA to RNA, known as a promoter region. And so that's how we know where transcription is supposed to begin. So we talked about yesterday that start codon, but upstream, so more to the 5' end of the start codon, that's referred to as upstream from the start codon, you have a sequence called a promoter sequence. And that's where RNA polymerase binds and where transcription is going to start. Okay, so that is within that promoter sequence, we have what's called the start point, and that's where RNA synthesis actually begins. Okay, so the piece of DNA that is actually transcribed, that primary transcript or pre-mRNA, is made from a piece of DNA referred to as the transcription unit. Okay, so that's the whole recipe that we're copying down here is the transcription unit. So we see here this transcription unit, okay? We've got our start point, and then this polymerase here is binding to this green little sequence in here that's called the promoter region. Okay, now that's of our DNA. Now, the way that polymerase binds, the orientation that it binds in, all of those things are important because they determine which strand is going to be the template strand versus the non-template strand. And which direction it's going to proceed in. So naturally, as we said, it's always going to go in the 5' to 3' direction. But, you know, the polymerase could, let's say, like orient itself this way, and it could read this strand of DNA. So the way that the polymerase binds and orients itself is really important. but it attaches to the promoter region, as we just said, and it starts at that start point, okay? And it's going in the 5' to 3' direction. That's the way that the mRNA is made. When we're reading the DNA on the template strand, the DNA is read in the 3' to 5' direction, just the same as replication. But the new strand that's being made, be it DNA in replication or mRNA in transcription, is always made in the 5' to 3' direction, okay? So this polymerase binds, and when it binds, it opens up kind of a DNA bubble, okay? And it unwinds that DNA and opens it up as it goes along. We don't have helicase in those enzymes here in transcription the way that we do in DNA replication. RNA polymerase is responsible for all of that. And so you see here, as it moves along, it's making its RNA primary transcript. It's opening up what it's needing to open in that small segment, and it's going to move along and make that transcript as it goes, okay? And at the end, it will reach a certain sequence where the polymerase will be removed from the DNA, and you'll have that completed RNA transcript, okay? So there's 3 steps here. You see them outlined. They're also going to be the same 3 steps in translation, so these are important. Initiation, elongation, and termination, okay? And again, we're going from 5, making it from the 5' to the 3'. We see here, end of the mRNA, meaning we're reading it from the 3' to the 5' end of the DNA. So this one here would have been the template strand. This one here would have been the non-template strand. I know that because it has to be read in the 3 to 5. So this one would be the 3 to 5, okay? And we're going downstream, okay? So this is kind of some new terminology with this downstream and upstream that we speak of. So let's go through these stages together a little bit more in depth. So initiation starts with some proteins that are referred to as transcription factors. You see them here in this diagram. If the transcription factors are not there, polymerase cannot bind, okay? The transcription factors must be there for polymerase to bind. You also have to have that promoter region for polymerase to bind. So together, the transcription factors, the polymerase, and the promoter region make up what is referred to as the transcription initiation complex, okay? Now, the promoter region is noted by a specific sequence of bases. It's referred to as the Tata box. I had this Indian professor, Dr. Sahi, at Western when I was in school, and I will forevermore be able to hear him saying the Tata box in my head. He was so descript when he talked about the Tata box. So that is what you have in the promoter region. That's what RNA polymerase recognizes and binds to be able to start transcription. And then also, like we said, these transcription factors, okay? So the Tata box is the promoter sequence. The transcription factors are the proteins. They are on the promoter sequence. And then RNA polymerase binds and gets going and does its job, okay? Oops. Maybe. All right. So that is the initiation step, okay? Now, the elongation step. In elongation, you have that RNA polymerase moving along, unwinding, as I just mentioned before, as it does, and using that base pair, that A to T, G to C, except we're taking A to U, and making the mRNA primary transcript as it goes along, okay? using the template strand. Notice this is the template strand here, and it's being read 3' to 5', which means the RNA is being synthesized 5' to 3'. Okay, this big gray blob is our enzyme polymerase, and the transcription is going in the downstream direction. All right, so that's initiation, elongation, and then the last is termination. Okay, so in bacteria, you have a specific sequence referred to as the terminator sequence that causes the polymerase to detach from the RNA and stop transcription. Okay, when it sees that sequence, it's almost, kind of like recognizing that stop codon. It stops, it seizes transcription, it falls off, and the mRNA is released. Now, remember we talked about yesterday that mRNA is not modified. In bacteria, there's no primary transcript or pre-mRNA. It's ready to go when it comes out. Okay, it doesn't have to do the modification steps. But in eukaryotes, we don't have a terminator sequence. What happens is when the RNA polymerase gets to a stop codon, it transcribes something that's referred to as a polyadenylation signal. And so that's basically a series of A's and U's, and then some proteins, some enzyme-based proteins, cut the RNA free from the polymerase and releases that primary transcript, that pre-mRNA, that then is going to undergo the processing before it becomes the actual mRNA that leaves the nucleus. Okay? So let's talk about this processing. We talked about it yesterday a little bit, but I told you we'd be going over it again. Sorry, I'm so congested right now. Before the mRNA is released from the nucleus, you have some additions made to it. So what you see here, here's our start codon, here's our stop codon. So the pieces in here is what actually code for a protein. Okay? Now, before the start codon, you have this part right here, the UTR. So that stands for untranslated region. And so that's kind of what the polymerase is working off of, an untranslated region. And then we have the polyadenylation signal. After the stop codon was reached, the polymerase makes a polyadenylation signal, and then it gets cut loose. Okay? After it gets cut loose, the RNA has to go through RNA processing, and that's what you see in yellow. We've got the 5' cap and the poly A tail. Okay? So the 5' cap is a guanine nucleotide, a modified guanine nucleotide, basically like some G's or guanines that gets put on the 5' end. And then the poly A tail is a series of A's that's going to go on the 3' end. Okay? And remember, I told you the importance of this is because that after the mRNA leaves the safety of the nucleus, it's going to travel through the cytoplasm where there's enzymes there. And enzymes like to cut, and they're going to try to cut and chop it up. And so it protects it from degradation in the nucleus. Okay? Now, in this, part that has been transcribed, we also have, I talked to you yesterday about introns and exons. So you see here, the dark red represents the exons. Those are expressed. They are coding sequences. They actually code for DNA. And then the introns are not expressed. Okay? So those pieces have to be cut out. So this is our pre-mRNA. It's had the 5' cap added. It's had the poly A tail added. And now those introns are going to get cut out. and that leaves a much shorter mRNA. So this is our final mRNA. That's not really what it's called, but it's not the pre-mRNA anymore. It's not the primary transcript. This is the one that's ready to go to the ribosome, okay? Now, what you'll notice is that it's quite a bit shorter. Most primary transcripts are about 27,000 nucleotides long, versus most proteins are about 1,000 on average nucleotides long. So we're going from 27,000 here to 1,000 here. So what you see is that there's a lot of introns, okay? There's a lot of non-coding stretches. And so those were spliced out. So let's talk about how they're spliced out. In this process, we have some molecules called SNRPs. That's SNRNPs, but we call them SNRPs. And they recognize the splice sites. These SNRPs come together with some other proteins to create a molecule called a spliceosome. And so these spliceosomes, made up of SNRPs, recognize the splice site from the introns to the exons, and they cut those introns out. So you see the cut out intron being released here, okay? And then they piece together the exons to end us up here with our final mRNA sequence, okay? Now, what's going to happen to this intron? It's going to be degraded in the cell. What cellular organelle is the trash can, is responsible for degrading things in the cell? Hopefully you said the lysosome, okay? And so that's going to be the job of the lysosome, to kind of take out that trash in the cell. All right. Now, the last thing that I want to talk about, this is the last slide, is ribozymes. So ribozymes are an enzyme, but they are an enzyme that is not a protein. All other enzymes are proteins, basically forever, okay? But ribozymes, hence the zymes, are enzymes, but they're not a protein. And so what they sometimes do is they can, in certain species, exist to, they work in the place of a spliceosome. They're an alternative to a spliceosome. And they can cut out their own intron sequences. Now, not all species have them, but certain species do have ribozymes, okay? And then the domains, okay? So we've talked about domains before in DNA. So domains are parts or polypeptides of a protein that are coded for by different pieces, okay? So we have one gene that has domain 1, 2, and 3. And those are kind of different polypeptide pieces that come together to make a functional protein, okay? They're separated by introns. And so that's what we have here. We have a domain 1, a domain 2, and a domain 3. And they can be coded for in one gene and make up one functional polypeptide, okay? So it's a piece of a protein coded for by a specific exon or a specific portion of a gene, okay? So that is all I've got for you on transcription today. Big player here was RNA polymerase. We've got the 3 steps: initiation, elongation, termination. We're going from 5' to 3'. We're cutting out the introns, pushing the exons together. That's the job of the spliceosome. Add the 5' cap, add the poly A tail, and send it on its way to translation. So when I talk with you tomorrow, we'll pick up with translation. Remember, transcription today happened in the nucleus, but tomorrow we'll be on the ribosome for translation. So if you have any questions, let me know. You all finish up the activity from yesterday. You have a Google Sheet or Google form with some questions just to review transcription today. And you all have a great day.